So in our syllabus, uh, we have so many topics in our syllabus, right? So let me read all the uh, topics. Let me read that. Yeah. So you have like determination of antigen and antibody reaction after Loney technique. So we have done with that, right? Then determination of human blood group antigens. This also we have done. Then raising of antibodies to specific antigen in the rabbit, that also be done. The racket immunoelectrophoresis that we have seen. And then determination of HCG by latex agglutination inhibition test. This we have not done. Right? This we haven't done.
Yes,
Then
sir.
ELISA we have done. Then a complete blood picture we have done. Then complete blood picture like it's like RBC count and then TLC. So these two will come in the same one. So that we have seen already. WBC differential count. WBC differential count you have to see. Okay. యాక్చువల్గా ఈరోజు మనకు డబ్ల్యూబీసీ డిఫరెన్షియల్ కౌంటే ఉండాలి కానీ వైట్ బ్లడ్ సెల్ కౌంట్ బట్టి డిఫరెన్షియల్ కౌంట్ ఓకే ద సేమ్ రైట్ దెన్ erythrocyte sedimentation rate we have done, packed cell volume we have done, and then determination of hemoglobin we have done, then mean cell hemoglobin and mean cell RBC volume now, osmotic fragility of RBC we have done. So only two things which are left over is like detection of HCG by the latex agglutination inhibition test is one. Another one is mean cell hemoglobin and mean cell RBC cell volume besides the white blood cell count, right? Differential count. Is it not?
Yes, sir.
So only those two topics is left out. Okay. So anyhow, Venkatech call chesara? Is he not available?
సార్ కాల్ చేసా సార్
Huh?
కాల్ చేసినా సార్ జాయిన్ అవుతాను అంట సార్
Okay, good. Yes. Okay, so the white blood cell count, or you can also call it as like a differential count, is one type of method, what we can say like a hematological method, which is essentially required for uh, to know the normal and the abnormal values of a individual person. Then what are white blood cells? How do we characterize them? So what are their characteristic properties? So their characteristic properties are like, whenever a like an organism or like a disease causative or infectious organism enters into the body, and the first thing which responds to that is white blood cells, which have a variety of ways by which they can attack. Like some will produce protective antibodies that will overpower the germs. So overpower the germs in the sense, so they, uh, like they attack the germs and then they will stop the activity of the germ. So when the germ activity is stopped, then automatically we don't get any disease, right? So some of them will produce the antibodies. Some of them will directly attack it. Okay, and the others will surround and endover the bacteria. So that means the organism or like a pathogen which enters into our body can be treated with the white blood cells in two ways. One is by producing the antibodies, so which is a little late process. So the antibodies will be produced later on. So the once the exposure is happened, so the antibodies will produce in a stipulated period. Whereas in the other way, the other white blood cells, they will be involved in what we can say like engulfing the pathogen, and so that it kills the pathogen. So they will just engulf ante mengadam, mingestai. So that means they surround it and then they will kill. Okay. So the white blood cells have a rather short half-life cycle, living from a few days to a few weeks. So the life cycle or the life period, half-life of red blood cells is 120 days. We know that, right? So that means the red blood cells can synthesize once in 120 days. That means every day we can see there are some red blood cells are getting synthesized in our body. But though they are getting produced, but you see that they will stay for like 120 days. That means for 3 months. Sorry, 4 months, right? For 4 months time, they will be in our system once they get synthesized. But in case of white blood cells, and we also know that the red blood cell number of red blood cells is more. The number of red blood cells are more, but whereas the number of white blood cells is very less. What do you mean by very less? So when we compare with the red blood cells, white blood cell number is less. So, and moreover, these cells are having short half-life. So when they have a short half-life, then in a few days of time or in a few weeks of time, so they'll disappear from the system, from the blood. Okay, so that's what it happens. And then several different and diverse types of leukocytes are there. We already saw this in the that is a blood picture, right? In the blood picture, we have seen what are the different types of leukocyte present, right? So, but they are all produced and derived from a multipotent cell in the bone marrow known as a hematopoietic stem cell. So they will be produced from, though there are different types of white blood cells appear in the system, but you can see that only of these cells are producing from the bone marrow, especially from the multipotent cells, which we can call them as like hematopoietic stem cells. So these are responsible cells for the production of leukocytes, all types of leukocytes. Leukocytes are found throughout the body. You can see everywhere they are there, in even tissues, even in muscle, even anywhere, any part of the body. So they are there. So the blood, the lymphatic system and other systems, so you can see the lymphocytes. All systems are responsible for, like lymphocytes are present, and then wherever there is an entry of these particular pathogens, then they simply attack. Okay. And the name white blood cell usually derives from the fact that after centrifugation of a blood sample, a white cells are found in buffy coat. You remember, right, in a yesterday class packet cell volume we discussed it, right? So in the packet cell volume, I told you, so the red blood cells will form as like a 45%, and that we called as like a packet cell volume. And whereas and afterwards in the above you can see there is a plasma which occupies 55%. But you can see in between the packet cells and the plasma, so there is a white buffy coat, right? You remember that, right? Do you remember this? What? You remember or not? I told you in the yesterday class. A centrifuge tube base tube ఇచ్చాను కదా మా నిన్న.
Yes, sir.
You remember that, right? So it's a middle layer, which is a buffy coat, white buffy coat, which we can see that is having a thin layer of nucleated cells between the sedimented red blood cells and the blood plasma, right? Which is typically white in color. So that's why the white blood cells are came as like, because they appear in white in color. And you also know that red blood cells are enucleated cells. They may not be having any nucleus, whereas the white blood cells are the nucleated cells. You see the basic difference between the white blood cells and the red blood cells, right? So the nucleus is present in case of white blood cells and nucleus is absent in case of red blood cells. And we know that the number of cells which are present in the number of cells present In case of red blood cells is more, and whereas in case of white blood cells this is less. And then the half-life of red blood cells is 120 days, and the half-life of white blood cells is like it's days to weeks, few weeks. That's what we can see. Okay, and then the scientific term a leukocyte directly reflects this description that the Greek derived from leuko in the sense white, and cyte means cell. So the leukocytes are nothing but white blood cells. That's what the leukocyte name indicates. Then what is the principle involved in the WBC count? So the white blood cells are nothing but WBC. How we can count them? So free flowing capillary or well mixed anticoagulated venous blood is added to a diluent. Diluent is like, if you want to add some water or something else, so added to a diluent at a specific volume in the thermopipette. So in the thermopipette, what happens? We are going to add a free flowing capillary or well mixed anticoagulated venous blood. So the venous blood is added to a diluent. So venous blood and ikkada. So we are going to take the buffery layer. Okay, and the diluent lyses the erythrocytes, but preserves leukocytes and then platelets. So what will happen here? You can see because you are adding it to the blood, right? A free flowing capillary or well mixed anticoagulated venous blood is having all red blood cells. That means like red blood cells, and then it has all types of white blood cells, platelets, and serum. Then when we add this diluent, so usually this diluent, what happens? It lyses the erythrocytes. So erythrocytes are red blood cells. So all the red blood cells are lysed, but you can see only the leukocytes and platelets were present. So they may not be get affected by this diluent lysis. Okay, lysis process. So the diluted blood is added to the hemocytometer chamber. So what will happen? So that means we are diluting it and then we are adding that into the hemocytometer chamber. What will happen? The specimen is usually EDTA anticoagulated blood because as and when you talk about like anticoagulation, so we have to go for EDTA addition. So we have to anticoagulate it. Then capillary blood is preferred. So usually we are going for capillary blood. So the capillary blood can be added to it. So the capillary blood in the sense you have to take only in a special pipette. That's what we mentioned here, like thermopipette. So the thermopipette is the one which is essentially required for the measurement of white blood cells, right? Okay, so we use anticoagulate EDTA anticoagulated blood, and capillary blood is preferred. And then the reagents and supplies which are required here, so including equipment, is like a white blood cell count diluting fluid we need. WBC diluting fluid we need, which may be one of the following, like you can see like acetic acid in the WBC. Acetic acid you can use. 2% acetic acid in distilled water can be used as WBC dilating fluid, and HCl 1% weight by volume can also be used as WBC dilating fluid. You can use any one of it. See, any one of these solution can be used as a WBC diluting fluid. So the acetic acid 2% or like HCl 1% or like Turk's solution, which is formed due to the addition of like glacial acetic acid 3 ml and 1 ml of crystal violet, and then distilled water 100 ml. Okay, so this can also be, that means the distilled water 100 ml in the sense it has to be made up to 100. So that total 100 ml is having glacial acetic acid 3 ml, crystal violet 1 ml. So that solution you can also use. Right, this is the thermopipette. You see here, thermopipette. You already know about this, right? Then white blood cells count dilating fluid we need. Then Thoma white pipette we need as an equipment. And hemocytometer is another one with cover slip is also required. Then microscope we need because the hemocytometer can be kept under the microscope to check the number of white blood cells which are present in the each block of the hemocytometer. So that we will also see. Then you can also see the lint-free wipe. A lint-free wipe is also required. So that means you have to remove some of the overflown or like the not required white blood cells or solution. And you also need alcohol pads. So these are the different materials and all equipment which are required for the WBC count. Then you know hemocytometer, right? So the hemocytometer is this. You see this one is the hemocytometer. You already have it in the lab. Even this one is hemocytometer. Here it is. And this is a Thoma pipette, Thoma white pipette, right? So this Thoma pipette, by using this, so we can suck this white blood cell solution and then that will be placed in the hemocytometer. So we are just sucking it and then we are placing it in the hemocytometer, right? So you can also have like a rubber sucking bulbs, tube, rubber sucking tube in the sense here you can have a rubber and then just you can press it and then you can suck the sample into the thermoball. So that you can see. Okay, let's see what is happening with the hemocytometer. So the hemocytometer is constructed. Okay, it's a counting chamber. You can say like it looks like it's slide, but it is a thick. Its thickness is high. You can see it's a very thick one. Then it's a counting chamber. So usually used for the cell counting, especially, and it is constructed so that the distance between the bottom of the cover slip and the surface of the counting area of the chamber is 0.1 millimeters. You can see it is constructed that the distance between the bottom of the cover slip and the surface of the counting area of the chamber is 0.1 millimeter. So that means only a thin layer of cells will be placed because if you want to count them, if you want to count the cells, then automatically what you need, you don't want, the overlapped cells. You want only the clearly dispersed, right, and distributed cells on the hemocytometer. So that's why the thickness or like, the space between the cover slip and the counting chamber is, it's only 0.1 millimeter. Okay. And the surface of chamber contains two square ruled areas. You can see there are two square ruled areas and which are separated by an H-shaped mat. Okay. So the H-shaped mat is always there. I'll show you that also. You see the surface of the chamber contains two squared rules. So you can see here in this one, you see this is two squared rules, right? But it looks like H shape. You see here H shape, right? So that's how we can see. That's what we can see with the hemocytometer. Here you can see it. So these are the two chambers. You can see this one and this one, and there are H-shaped square. You can see the mat. Okay, mat. Okay, that we can see. Okay, so what we'll do here, let's see. These are the counting chambers. These two are the counting chambers and properly load the sample here in these two places. And the cover slip is this one, you see, it is covering completely from here to here, you see this one. Then you can see the cells are looks like this, you see. So the flooded, that means when we place the cover slip and the excess amount of fluid will come back, will be going to the outside. So that also you can see. And then underloaded, you see that less amount here and it is fully loaded. And then you can also see some air bubbles here. So that also interferes the counting of the white blood cells. Then the procedure for counting of white blood cells is like draw the blood up to 0.5 ml mark in the thermopipette. So that means we have to take thermopipette and draw the blood. So take the 100, the red blood which is just obtained, or you can also say like EDT added blood you take, and then 0.5 mark up to 0.5 mark. So using thermopipette, you have to suck that blood. Then wipe the outside of the capillary pipe. So we need wipes, right? So you need wipes, and then the wipes can be wiped in the outside of the capillary tube, which there is some blood is stick to that uh, thermopipette, right? So the thermopipette will be cleaned in the outside with the what we can call it as like wipes. Okay. So if you don't wipe, and then you can see that there is an interference of this uh, blood with the WBC count. Okay, so that's why you have to go for wiping of the all the blood molecules from the thermopipette. So holding the pipette almost vertical place into the fluid, draw the diluting fluid into the pipette slowly until the mixture reaches the leaven. Hold the pipette almost vertical place, right? So it's not horizontal, it's vertical, straight, up straight, right? And then into the fluid. So then place into the fluid. So that means the fluid is like WBC dilating fluid, and draw the dilating fluid into the pipette slowly until the mixture reaches the level mark, because the pipette is already having 0.5 mark varkuna blood is there, right? And now from the blood, what is happening is we are sucking the by placing this 0.5 mark thermopipette in the WBC dilating fluid, we are going to suck the WBC dilating fluid until the total volume reaches to 11. So that means it is already 5, and then 6 more marks, 0.5 and 0. So 11 mark, until 11 marks we are going to dilute while gently rotating the pipette to ensure a proper amount of mixing. So you have to rotate it properly, and so that you can see that the gentle mixing of the sample. So the sample, in the sense the diluting fluid as well as uh, all the white blood cells and red blood cells will get mixed with this diluting fluid. But this diluting fluid is responsible for killing, lysing the red blood cells. So it kills or it lyses red blood cells. So when the red blood cells are lysed, then automatically the leftover cells are the white blood cells, especially different types of white blood cells will be there. And also you can see what is that? The platelets can also be absorbed, right? So that's what we can see. Then they place the pipette in a horizontal position. So when we are taking the diluted fluid, WBC diluted fluid, and we have to keep that pipette, the thermopipette in the up straight, right, vertical position. And once we draw up to 11 mark, and then you have to keep that in a horizontal position, right? And firmly hold the index finger of either hand over the opening of the tip of the pipette and detach the aspirator from the other end of the pipette. Now the dilution of the blood is completed. So that means we have to remove all these parts of the. So you see here, this is 0.5, but you can see that there is 11 mark is here, right? Up to here. Then you can detach this tube from the thermopipette. Okay, so that's what we can see. So place the pipette in a horizontal position and firmly hold the index finger. That means you have to hold it strongly without disturbing the pipette. Either hand over the opening the tip of the pipette. Detach the aspirator from the other end of the pipette. Now the dilution of the fluid is or blood is completed. So now we'll go for mix the sample for at least 3 minutes to facilitate homolysis of RBC. because I told you already, diluting fluid can homolyze the RBC, and leftover cells are WBC and platelets. And clean the homocytometer. So you have to take the clean homocytometer and its cover slip with an alcohol pad and then dry with a wipe. So that means you have to make sure that you are cleaning or the chamber and everything is clean. There is no interference of previously founded cells in this experiment, right? So in that case, then you have to load before filling the chamber. Discard the first 4 of the 5 drops of mixture. So you just remove the first 4 and 5 drops of mixture. Don't use that for counting. And then on a piece of gas to expel the dilute from the stem. So that means the dilution will happen here, effective dilution. The stem is this one, bulbuli structure in the that is the one which is a stem. So when we mix it, so then mixture will make the red blood cells to lyse properly. But you see, when the solution is in this capillary, you see here, and that may or may not be like the red blood cells. So in order to remove this, what we call it as like capillary blood, so we'll first 4 or 5 drops of blood is removed. So when it is removed, then automatically from here to here, all the blood cells or the blood which is not properly mixed will be common. So now once we release the 1 to 5 drops or 5 to 7 drops, then automatically the solution which is there in the bulb stem can reach us to the tip of the therma pipe. Okay. Then it's more important that so you have to use the what we can say before filling the chamber, discard the first 4 to 5 drops of the mixture on a piece of gauze to expel the dilution from the stem. Then carefully charge hemocytometer with diluted blood by gently squeezing sides of reservoir to expel. So gently you have to add this. So spilling ఉండకూడదు. So it cannot be spilled out. Okay. And the contents until the chamber is properly filled. So you have to add all the contents of the the therma pipette into the chambers of the hemocytometer. But while doing it, you should not be spilled. Then so then you have to take that hemocytometer. Of course, you have to place the cover slip and then you have to go for examination of the WBC. So under 10x magnification, scan to ensure even distribution. Leukocytes are counted in all 9 large squares of counting chamber. So leukocytes are counted in all 9 large squares of counting chambers. Okay, so large counting chambers are there, four chambers are there, that 9 large chambers are there, but you have to use each chamber for calculation. So you can calculate like, you can choose in each chamber, you can choose like 1 square or 2 squares or 3 squares randomly, and then you can count the number. So the move the upright corner square, bottom right corner square, bottom left corner square, and end in the middle square. So you have to look at the different white blood cells which are present in that. So the count all the cells that touch any of the upper and left lines, and do not count any cells that touches a lower or right line. Okay, so this is how you have to count it. You see here. So the chamber looks like this. So you can have all this, you see. This is the portion where we can go and read the cell number. So we are going to read all 4 corners of the middle shell, right, which is already subdivided into some other shells. And then you can also see the middle one, you see. So this is having like 5 major divisions, right? But This side also there are 5. That's why we will go for like a middle shell, right? This. You see, when we go for like this, so this will be like this will be like this, and this will be like this will be like this. So you have to be very sure that you are going to calculate it number. Then from that you can calculate it with a formula. You can dilute or you can see the calculation. So the depth is usually we know that 0.1. I already told you that is how the power slip is fixed, and so that the cells will be distributed even. The correction for dilution. So how do you correct the dilution? The thermopipette is like 1:20%, and dilution factor is 20. So because you are diluting 20%, because you are taking only 0.5 0.5% of the blood into the thermopipette, but you are diluting it up to 11, right? So that's why the dilution is always like 20, 1:20%. అంతే కదా? See, because you have like 0.5 ml and you are going to add 10 ml. Okay. So 11 and 10 కదా 10.5 ml ఉంటుంది. So what is the meaning of 10.5 ml? 5.5 ml ki 10 ml అంటే it is 20 times. 0.5 ఉంటాయి అందులో, right? That's why it is diluted 20 times. So the dilution factor is 20, and correction of volume is another aspect which we have to do. And volume of first small square is like 1 into 1 into 0.1, that is equal to 0.1 millimeter cube. And the volume of 4 large squares is like 4 into 0.1. So that's what we can see. So the 0.4 millimeter cube or microliters we can call it. Suppose that you count 50 cells in 4 squares, 0.4 millimeters in 3, and then found the count in 1 millimeter cube, right? Then that is equal to 50, which gives the 0.4 millimeter cube, and the x we don't know, so that's like 1 millimeter cube, and x is equal to, for example, x is equal to 50, and 50 into 1 by 0.4 will give us the value. So the volume correction can also be done here. So the volume correction can be done by like 1 by 0.4%. Then the total count of cells per 1 millimeter cube are always calculated using the formula number of cells into volume correction into dilution. Number of cells into volume correction into dilution. So that is equal to number of cells into 1 by 0.4 and into 20. So based on this formula, we can calculate the number of white blood cells which are present in the cell. A high elevated leukocyte count may make accurate counting difficult. So a high elevated leukocyte count. So if there are like a more number of leukocytes, so that can be called as like leukocytosis, and it may make accurate counting difficult. So we may not be able to count properly. So when there is a high amount of leukocytes, and in either instance, the secondary dilution should be made. So that's why you have to go for like secondary dilution also. When the secondary dilution is done, then automatically you can see there is. You can compare the samples, and when you compare the samples, then automatically it helps us whether we have done properly in the first or the second. So that is how we can do that accurate count. Okay. In either instance, a secondary dilution should be made, and when calculating the total account, it should be adjusted to the formula to allow for secondary dilution, or the red pipette can be used to mark 1:100 dilution. So not 1:20 dilution. So you have to go for 1:100 dilution. So you, in general, 1:20 dilution is more than enough to count the RBC. Sorry, the WBC. But whereas when the high number of cells are present in the in the blood, then automatically you have to go for like 1:100 dilution. So You need to go for large amount of dilution. Otherwise, the cells will be overlapped and then you can't see them. You will see them, but you can't count them. That's a problem. If count is less than 3000 cell per millimeter cube, a smaller dilution of the blood should be used to ensure a measure accurate account. So it accurates the measure accurate account, and this can be accomplished by drawing the blood up to 1 mark and the diluting fluid to the 11 mark, and the dilution will then be 1:10%, and the dilution factor in the calculation will be 10. So that's how we can see that you can increase the cell point out, or you can say like the cell, what I can say the identification under the microscope. And if more than 5 nucleotides, RBCs are seen on the differential, the total leukocyte count should be corrected using the following calculation. So that means if you want to count the differential count is always like, there are different types of leukocytes, 5 types of leukocytes like macrophages, mononucleated, dinucleated white blood cells, or like uh, the other leukocytes, right? So these are all other leukocytes. When we calculate all these like binucleated, uninucleated, and so on. So this will help you. Okay, so this will help you to find out. So the differential count we can go for, and that differential count will help you to see what are the different types of white blood cells which are present in the blood. So for that, we should go for like corrected WBC is equal to uncorrected leukocyte count into 100, uncorrected leukocyte count into 100 by 100 plus a number of NRBCs or 100 WBCs on differential count. So this we can see, and then you can substitute all the values if you have, and then you can calculate the corrected WBC. So that means total amount of WBC molecules can be counted. What are the common sources for the failure of the WBC or like blood volume? So the failure to have required blood volume. So common sources of errors we can see. Failure to have required blood volume, failure to mix well. So that means if the required blood volume is not there, if you fail to take them, then automatically it will become an error. So you'll get a different value, but not the exactly the same person's value, right? So that means the white blood cell value in the person. Then failure to mix well. If you are not able to mix, or if you are properly not mixed, then it can also lead to the failure of the WBC count. Then the failure to discard the first 4 drops. That also I told you like so here what happens, stoma pipette needs to be what we can say thorough mixing. Thorough mixing will be done only in the stem but not in the capillary tube. So that's why the capillary tube blood will be removed first 4 to 5 drops, and then we'll go for the next solution will be placed in the hemocytometer channel, right? So the 4 drops should also be discarded. So that's what we can see. Then next is failure to properly change the counting channel. So you have to be very sure that you are changing the counting chamber and cleaning the counting chamber. Otherwise, you can see the overlappings, or even the last time cells can also be appear here. The least frequent sources of errors which we can see in the WBC count are inaccurate pipette or counting chamber. So if the counting chamber or the pipette is not accurate, then automatically you can see there is an error. Then the moist or unclean pipette. So you have to use only the cleaned pipette which should have moisture. Okay, so that pipette can help us. If not, and then we can see that there is a damage in the test. And then excessive pressure in finger while obtaining the blood. So you have to use the pressure while obtaining the blood so that what will happen? We'll get the more number of circulating blood. So that's why that is how the blood will be taken out from the persons, right? Too little or too much dilution fluid is also not good. That means you should not dilute it highly, or you should not. What we can say that the high dilution is banned, and then low dilution is also banned. So that can also affect the red blood cells. And slowness in manipulation. So I already told you like how do we manipulate the different solutions and also that if you do slowly, then that also can create a problem. Okay, slowness of the manipulation, thus allowing the blood to clot. So which allows the blood to clot, and then you can also see like air bubbles in the pipette can also cause damage, can also give wrong results. Then air bubbles in the counting chamber is also give some, will also give some errors, right? Then presence of yeast or other contaminants in the diluting fluid is also another aspect which we can see. The presence of many nucleated red cells causing a high white cell count, that is also another aspect which we can see. Then mistakes in the counting or calculations. So if you are not properly able to identify under the microscope, if you are not able to see the cells in the microscope, and then if you are skipping the cells in the microscope, so that also leads to the improper counting. So that means we'll get the wrong results in case of white blood cell count. Okay, so this is what about the WBC count. Do you have any doubts in this? Any doubts, ma'am?
No, sir.
Yes. Maybe we'll also see the differential count also, right? Okay, so in 3 more classes, hopefully I'm going to complete all this, what I can say, like practical classes also, right? So the practical is nothing but we can say it's a, what I can say, it is also a theory class now for you. So the only thing is you are just knowing what is what, and that you are going to count, and then that will help you. That means these procedures can help you to do the practicals at your laboratory, right? Are you clear?
Yes, sir.